demonstration how to remove the gonad from a grouper, how to sex them if they're sexually active, and how to do a few other things in terms of helping us out with our grouper studies. Those of you that might be collecting fish out in the Gulf and then bringing them back in and collecting samples for us. Now this is a yellowfin grouper. It's common in the Bahamas. We're over in the Bahamas filming this, Microperca venenosa. This is species is very close to what you call the black grouper in the Gulf, or the gag grouper. It's in the same genus, and so basically what I'm showing you here for this yellowfin grouper will be directly applicable to the gag, or black grouper as they call it in the Gulf. And a little bit later I'm going to show you a Nassau grouper, which is similar to the red grouper in the Gulf. Okay, I'm going to go ahead and open up the body cavity. This fish was brought out of about 80 feet of water. The swim bladder's a little bit blown up. It's inflating there now. Okay, I've opened up the body cavity, and okay, why don't you stop there now? You're going to have to get rolling. The belly of the fish. Now, ideally for collecting the gonads, you shouldn't slit the fish along the midline here, because you're liable to damage the gonad as you cut through it. Cut through on one side, and the most important thing to miss down here where the anus and the genital opening is, you want to miss that. So go to the side and around that. Then you can open up the body cavity. You can see inside here. We can see various parts. This is the swim bladder up here. It's still inflated. You can puncture that, let the air out of that. We have the pyloric ceca, the stomach right here, and then the liver up here. Now the gonads on both the male and the female lie just on top of the intestine. They'll lie basically up on the top of the body cavity right below the swim bladder, and they will actually come down and connect right about where the intestine comes down and connects at the anus of the fish here. And your gonads will be a paired structure, and this fish, unfortunately, it's not very sexually active, so the gonads are not real large. But this is the two gonads right here, the two lobes of the gonad, this yellowfin grouper. I'm going to go ahead and dissect these out now. And now if you had a fish that was in spawning condition, these gonads would perhaps be as much as that long and the size of your fist. So size isn't really very indicative of what is or isn't a gonad. If you're collecting gag during March or April, they're going to have really large gonads, and they're going to be basically the size of your fist when you open the fish up. So if you were collecting a sample for us, what you would do is, first of all, measure the fish. Okay, we're interested in the total, or sorry, what we call the standard length of the fish and the total length. But the most important thing you can give us is what's called the standard length, which is actually the distance from the snout to the end of the vertebral column.
Hold on a second, I can't get all that in from this.
Yeah, okay. All right, the first bit of information we're interested in is the length of the fish. In other words, how big is your fish? It's not that important that you actually weigh the fish for us, because we can convert from the length to the weight using tables and such that have already been developed. So all you need to do basically is measure the length of the fish. And there are various ways this is done. First of all, you can use what's known as the total length. In other words, that's the length from the snout basically to the farthest point of the caudal fin here. That's okay. The only problem with that is that if the tail is damaged or something, that can vary the actual total length of the fish in fish of what are actually the same weight or the same size. A more accurate method of determining the length of the fish is what we call the standard length. Okay, and that's basically the distance from the snout to the end of the vertebral column in the tail. Now, you may not know, may not sound very easy how to determine where the vertebral column ends. It's actually very simple. All you have to do is take the tail of the fish and bend it up, and you can see in essence, a line which appears right here. Just bend it up, and there's a line where the tail bends. That's the end of the vertebral column, this point right here. Okay, so this, actually from here to here, is the official standard length of the fish. Now it gets a bit difficult, some of the ways that you measure these, actually measuring from this part of the snout to the tail. With things like gag and this yellowfin grouper, we actually go ahead and just measure from the tip of the lower jaw. So you can do this with a tape. In other words, take a tape measure, stretch it across here from here to here. There are measuring boards that fisheries biologists use to measure this sort of thing also. Okay, I'm going to stop this. All right, now if you're just measuring this fish, say with a yardstick or something like that, what you'd basically do is just, hold your straight yardstick as close as possible to the snout there, the tip of the lower jaw, so you can get it as accurate as possible. And measure back to where we said the end of the vertebral column is, right about there. This particular fish measures out at 72.5 centimeters, or about 28 3/4 inches. Now you can measure it either in inches or centimeters. It makes no difference to us, just so you measure it accurately as much as you can. Now if we were going to measure this fish on a measuring board, what you basically have is a board here on which a ruler like this or yardstick is mounted, and then it will have a vertical piece of wood right here. So you can set this down and just bring your fish up, lay it on, push it up until the upper or lower jaw hits that board up here, measure or bend the tail up like I showed you, and then just, I usually just hold the knife here, and you can see what the actual length was. So that says like 28.5 inches. So you can measure fish very rapidly. If you've had a real good day and got 50 grouper out there or something, you can measure them in about 10 seconds a piece using a measuring board. Otherwise it takes longer if you measure each individual one. If you have a measuring tape, measure it as accurately as you can. There is a little bit of error if you go across the curvature of the body, but it's fairly small. The most important thing you can do once you've measured the fish is you've got to write the data down. If you don't write it down and keep things squared away one way or another, it gets really confusing, and it won't do us any good in trying to understand the biology of these groupers. So I'm going to write out a label here for this particular grouper. Okay, this is a piece of special label paper which won't fall apart when it gets wet. I'm going to use just a standard pencil to record the data with. If you use a ballpoint pen, it'll just evaporate or just melt off the paper. So you want to use a pencil preferably to write the data down. So the information we need, first of all, we need to know what kind of fish this is that you're sampling. So this is a yellowfin grouper. Okay, yellowfin. Now it's very important also to know the day you collected that fish. So today happens to be the 26th of October 1989, and it's also of interest the time of day where you collected this fish. We collected this fish about 10 a.m. this morning. Okay, so now we have the species we're dealing with, the date it was collected, and ideally the time it was collected. All right, now I've taken one piece of data looking at the length here. I can write that down on here also, so all the data is in one place. Okay, that was 28.5 inches, standard length. You have to say, what that length is. Is it standard length, which I've abbreviated here as SL, or is it total length, which would be TL. But write down the length, the measurement, however many inches or centimeters it is, and then what type of measurement it is. If it's standard length, is it total length. Okay, kill it for a second. Okay, I'm going to go ahead and remove the gonad from this fish here. Remember I said that's this paired structure. There's one half, there's the other half. Found down near the anus, it'll be on the upper side of the intestine usually. It's attached by membranes. You see these clear sort of membranes here to the lower part of the swim bladder. Okay, all you have to do is cut these membranes out. I'm sure most of you have cleaned a lot of fish in your time and will be quite good at this, but basically all you have to do is slice those membranes out, and you'll get down, as you get down to where it actually empties out at the genital opening, you'll get down where you have the paired gonads here attached down here by just some sort of a tubular tissue-like structure. Okay, so cut them free of the membranes, and all you have to do is go ahead and cut them off essentially at the skin. Okay, so you'll end up with paired gonads like this. Interestingly enough, when fish are not sexually active as this one is, it's very difficult to tell males from females. I can't be really sure even which this one is, but those are definitely the gonads. When they get a bit bigger, and we might be able to see this in the Nassau grouper we're going to cut up next, you'll be able to tell the males from the females, but that's the gonad there. This is what we are interested in as a sample. What fisheries biologists do basically is they compare the weight of this, the weight of the gonad, to the weight of the fish, and come up with an index that tells us how whether the fish are spawning or not. We're also interested in the structure, how many eggs are in the gonad of the female, and a lot of other different things. So this is basically what we want you to save, just the gonads. All you have to do now, okay, is take that gonad. There'll be separate plastic bags for each gonad. You can use bags we might give you, or you can use plastic baggies or anything you have. You could wrap these up in paper towels if you had to. The most important thing is to keep the gonads separate from one another and with the data that goes along with them. So I'm going to put these gonads in this plastic bag here, and I've got the label that I made up here. It's got the species, the date, the time, the length. If I know what sex this is, you know, say if it's a female, I can go ahead and write that down on the label. Anything else I think that's of relevance, but this is the basic information we need, and that should go in the bag with the gonad. Seal your bag up somehow. You can tie it in a knot, or use a little bag closer or whatever. If you've got Ziploc bags, just zip it shut. Okay, that part there, that's the sample. That should go in your refrigerator or in your freezer. It's perfectly okay to freeze them. And you can go ahead and clean the fish like you would normally, but this is what we'd like you to save for us. And then we'll pick these up from you. We'll actually go ahead and weigh the gonad later on. From the length, we know how much the fish would have weighed, and we'll come up with our calculations about how much the gonad weighs relative to the fish. Okay, next. This is the next fish we're going to do. This is a Nassau grouper. This would be very similar to a red grouper in the Gulf. It's a member of the genus Epinephalus, very similar to your red grouper. This hopefully will have a bit better developed gonad at the present time. One thing I didn't mention when we were talking about cleaning this yellowfin grouper is it's also important to know the locality where the fish was taken. And so we'd ask you that on your label you write down more or less where it was taken. We're not interested in getting anybody's Loran numbers, but just a general idea where the fish was caught, like 20 miles off St. Mark's or 25 miles. And the water depth would also be of interest to us, either in meters or feet or fathoms, anything, just as long as you specify how many of any individual unit water depth that you caught it at. So just a general idea where the fish was caught, so we know if it was caught inshore, offshore, that sort of thing. Okay, well, I'm going to go ahead and open this Nassau grouper up and take the gonads out of it. Now remember, instead of cutting right along the midline, we're going to cut it up on the side. You can do this on either side, the left or the right, but basically you won't damage the gonad too much cutting it up here. Why don't you stop it for a second? This Nassau grouper opened up here. Looking in again, you can see here's the liver, the pyloric ceca, part of the stomach. Here's the intestine down here, and here we have the gonad right here, laying up on top of the intestine, right below the swim bladder up here. This one's quite a bit better developed than that yellowfin we just did. So I'm going to go ahead and measure it now, and then we'll go ahead and take the gonad out. Now notice on, like this will be the same on red grouper, on this Nassau grouper, the lower jaw doesn't stick out as much. It's a little bit different than your gag and your yellowfin. But just go ahead and measure from either the lower or the upper jaw. And then back here, remember, just bend the tail. You can see where it bends easily. That's the end of the vertebral column right there. Okay, that comes out to 26.5 inches from here to here. If I measured the total length back to here, that's about 30.5. So you can see there's a 4-inch difference between total length and standard length. Remember on your data thing that goes in the bag that you have to write down which type of length it is, whether it's standard length or total length, so we don't get confused. So making out the label for this bag again, this is a Nassau grouper, 26 October 1989. Now we caught this off Darby Island here. We're in the Bahamas. Darby Island, Exumas, Bahamas. And the standard length was 26.5 inches, 26.5 inches. Okay, so I'm going to go ahead and put that aside. Go ahead and remove the gonad here. Just like in the yellowfin there, these are connected by a membrane that you see, maybe you can see here in the picture. Just slice that membrane off. Don't cut yourself in the process. Okay, and when you get that pretty well removed, there's the gonad there. You can see again it's 2 lobes. It's 2 lobes to the gonad. This one is probably a male. I can't be 100% certain, but it's probably a male. Now we can look at this under the microscope even at this stage and tell whether or not it's a male or a female. And when you're out in the field collecting fish and sampling them for us, you don't have to really worry about whether it's a male or a female. We'll be able to decide that once we see the gonad that you brought back to us. Okay, remove that, and there's the gonad from this Nassau grouper. Okay, I'm going to put that away in the plastic bag, put the label along with it, put it in the freezer. We're all set with this one. Now, one other thing that we're interested in that some of you might be willing to help us with, we're also interested in collecting the ear bones from groupers. People use these to age groupers by looking at the rings in them to see if how many years old they are. So with a fish like this, by knowing the length and the age, and if you've sampled a large number of individuals, you can get an idea of the growth rates of a whole population by sampling these otoliths. So it takes a bit more time to take an otolith sample. However, if anybody's interested in doing it, it would be quite valuable to us to be able to get more information about otoliths and the growth rates. Now I'm going to show you how to take out the one of the pairs of otoliths in this Nassau grouper here. The yellowfin and the gag grouper are a bit more difficult, and I'm not going to demonstrate that, but go ahead and show you in the Nassau here. Basically, the ear bones are in the inner ear of the fish, which is on the lower surface of the skull. So we can either go in from the top, which is very difficult, or we can sneak in from the bottom of the skull and get those ear bones out. So I'm going to go in from the bottom here. It's one of these sort of things, once you know how to do it, it's very easy, but sometimes learning how to do it the first time is a bit difficult. So we're going to be going in, turning the fish so you can see it here, into the bottom of the skull. I'm just going to cut the gill cover a bit so we can move it out of the way. Okay, that exposes the bottom of the skull a bit better. Now the gill arches here are in the way. I'm going to have to get them out of the way also. So what I'm going to do, this would be just like gilling the fish in general. I'm going to cut them out, starting up at the top first, removing them. You've got a good, sharp, sturdy knife. This is pretty easy. Why don't you stop it there? Removed on this side now, and we'll go ahead and cut the otolith out. Now there's a pair of otoliths, there's one on either side of the bottom of the skull, so we're going to go ahead and take one out now. Now in this epinephrolus, the upper part of the gill arch attached right in here. You can see this flesh sitting right here where the muscles where it had attached. If you get your finger in here and feel, there's a bump right there on the bottom of the skull. So the ear bone is right inside there, the otolith. So what I'm going to do is cut the tissue out of the way here so we can get down to the bone. Okay, I think you can see this here in the video. There's a white bone here. That's the bottom of the skull. The otolith is right inside there. All we have to do is take our knife and cut into that carefully. You don't want to cut too deep right away because you don't want to damage the ear bone, but cut into that. You want to kill that, so I gotta sharpen this knife. Top of the thing with the otolith here. Just cutting away at the bone. And if you can see right now, we're coming to a chamber in here, right in there where the knife tip is.